Hello friends, in this session I will be give that detailed way in the with the chemical structures of the various intermediates, what are the involvements are there in the citric acid cycle. In the previous video I given that glycolysis in the detailed way with the chemical structure, how it has been taking place in the cytoplasm of the cell, the glycolysis and converting glucose into pyruvate in the previous video. Now let us come to this session, the citric acid cycle. The citric acid cycle, another name TCA cycle, another name also there, citric acid cycle, and because the name is tricarboxylic acid cycle, another name also tricarboxylic acid cycle. So we are observing here the three molecules of the COOH group. It is a first product in the citric acid cycle by the combination of oxaloacetate with the acetyl CoA, and we are getting the first product in the form of citrate. So here the based upon the arrangement of the writing, if we are ionic form, if we are writing, we should be called citrate, otherwise citric acid. And here also all these intermediate compounds based upon the written part in molecular levels, we should be announced. So suppose here also oxalosuccinate means 8 form we are showing, that's why. Otherwise if we are keeping H, oxalosuccinic acid like that, we have to pronounce all these areas. So now let us come to the citric acid cycle. That will be tricarboxylic acid cycle, another name citric acid cycle, another name TCA cycle, and it has been found by the Hans Krebs in 1937. The nematodes and Pigeon mussels. He found the great scientist, and he got the Nobel Prize in 1953 also. He got Nobel Prize by the Hans Krebs, and that's why you are also calling Krebs cycle. So here the different kinds of names also we are calling, and even in the competitive exams also they may be asked different names, either Krebs cycle or citric acid cycle or TCA cycle or tricarboxylic acid cycle, all are same cycle. So we should be remember the different kinds of names for this cycle, and very important cycle also. This will be taking place in the matrix of the mitochondria in the eukaryotes, whereas in the case of prokaryotes it will be taking place in the cytoplasm. And here first of all, first we have to discuss one by one in the stepwise manner. So first we have to take this pyruvate. The pyruvate is the end product of the glycolysis, and after pyruvate, after getting from the glycolysis, the pyruvate converted into acetyl CoA. So for this formation, and that this acetyl CoA will be entered in the matrix of the mitochondria, and how it will be converting the pyruvate into acetyl CoA. So here pyruvate dehydrogenase complex will be very important to convert the pyruvate into acetyl CoA. So here structure CH3C11O COOH. So 8 form we are showing, that's why pyruvate. If we are adding here a COOH group, we should be pronounced as pyruvic acid. So like that, all these things we should be remember. Either pyruvate 8 form or acid form, like that. So based upon the written. So here the pyruvate converted into acetyl CoA. Here coenzyme involvement is there. Is CoA also adding, reducing equivalents also adding NAD plus, and we are getting NADH plus H plus, and CO2 also liberating. And in this pyruvate dehydrogenase complex, we are getting the dehydrogenase enzyme and decarboxylase enzyme and transacetylase enzyme along with Mg2 plus also required, and TPP, transfer thymine pyrophosphate also required, and lipoic acid also required. So these are all helpful. These total factors, we should be call it as pyruvate dehydrogenase complex. This complex will be helpful to convert the pyruvate into two acetyl CoA molecules. And one more important point here is the pyruvate. So we are getting from the proteins. Proteins nothing but the combination of the amino acids with the peptide bond. So here the oxidative catabolic pathways. So interlinking is that acetyl CoA, the central part, it has been interlinked to the fat oxidative catabolism as well as the protein oxidative catabolism also. So here and the proteins converted into amino acid by the oxidative catabolic process, and here the amino acid, one of the amino acid alanine. So from that alanine also we are getting the pyruvate, and these are the interlinking pathway. It is a amphipathic pathway because of the involvement of the synthesis as well as the catabolic process. Both have been involved in this amphipathic pathway. That's why we are calling amphipathic pathway. So we are calling. So here both involvement of the catabolism related, anabolism related. This will be interlinked to the fat oxidation as well as the protein related degradation purpose also. And many more intermediate compounds also helpful to synthesize some of the intermediate compounds or amino acids also. So that will be, so that's why we are calling amphipathic pathway. That is nothing but citric acid cycle. So here, so after acetylation process, dehydrogenase also helpful, and decarboxylation also helpful, and transacetylase also helpful, and Mg2 plus also helpful, thymine pyrophosphate as well as the lipoic acid, and lipoic acid also required. So in this pyruvate dehydrogenase complex, and that will be converted, you see that CH3C11 goes COO8 form, that will be converted into CH3, C11O, C11O. So here this will be in the form of CO2. It has been releasing out the two molecules. And here are the two molecules of involvement of the NJDs, and we are getting two molecules of NJDH plus H2 plus. And each NJD, nicotinamide adenine dinucleotide, releasing 3 ATPs. Now if you are multiplying two molecules, that will be, you are getting 6 ATPs molecules in the pyruvate converted into two molecules of acetyl-CoA. So here that SCOA coenzyme adding to the reaction, so that's why instead of this CO2 will be removing, and in this place the SCOA, acetyl-CoA, the coenzyme, it has been adding in this place. So that's why in the each and every steps we need to compare the previous one to further product whatever we are getting. So that's why you can understand easily in the chemistry point of view in the detailed way, and how that rearrangement of the molecules taking place. And here this will be the first reaction. So in pyruvate converted to acetyl-CoA with the help of pyruvate dehydrogenase complex, and after formation of this acetyl-CoA that has been entered into the matrix of the mitochondria, and afterwards here, and also this will be interlinked to the fat oxidative catabolism. So fat, it is a very lengthy fatty acids. We have saturated fatty acid and unsaturated fatty acids based upon the double bond, whether it is present in the double bond, that will be unsaturated fatty acids. Example, linoleic acid, linoleic acids. And the formic acid is the saturated fatty acid, 16 carbon containing fatty acid. Like that, based upon that fatty acids, whether it is double bond is there or single bond is there, based upon that saturated and unsaturated fatty acids are there. Now let us come to in this point the fat oxidative catabolism through the beta oxidation process also happening, and we are getting two acetyl-CoAs. From that acetyl-acetyl-CoA rearrangement, and it will be forming, and finally we are getting the acetyl-CoA. That is also one of the link, and this is also one of the links. So protein link also there, and fat link also there. So carbohydrate link also there. It is a total connecting link of the carbohydrates, the fats as well as the protein related degradation and synthesis process, and many more involvements also, the intermediate compounds, and it is one of the energy rich compound. The acetyl-CoA is one of the energy rich compound we got, and afterwards it has been entered the matrix of the mitochondria, and it is an aerobic process. It will be required oxygen. That is an aerobic process. So now comes to the here the oxaloacetate. So here that COOH, CH2, C11O, COOH. That is a 8 form we are showing like that. So that's another one, oxaloacetate. If we are writing like that in any form, acetate we should be pronounced. Otherwise, oxaloacetic acid. So we should be pronounced. Now here oxaloacetate combined with the acetyl-CoA, CH3C11O-S-CoA, and here COOH and CH2C11O-CO. So here we should be, these two are combining, and we are getting the product citrate. And this is the first product in the PCS cycle. This is the cyclic process. That's why you are calling citric acid cycle also. So here, this will be the 1, 2, 3, 4 carbons containing oxaloacetate combining with the how many carbons? The 2 carbons of the acetyl-CoA with the help of citrate synthase enzyme. Synthesis, which product? And the citrate is the synthesizing. That's why the enzyme name also. Age will be common. Each and every enzyme and age will come. That should be identified as enzyme. So that's why the citrate synthase with the combination of oxaloacetate with the acetyl CoA, we are getting the citrate. So citrate, here the 6 carbons, 1, 2, 3, 4, 5, 6 carbons containing citrate we got. So here that water molecules also involvement is there, and HSCOA will be moving out, whatever present in this one will be going out, and you just compare that structure COOH and CH2, and here the CO, and once again it will be adding here, and slowly C double bond O ketone rearrangement, molecules of rearrangement happening, and finally we got these two after combination we are getting the citrate. So now the citrate is the 6 carbons containing the first product we are getting in the TCS cycle, and next the citrate, and some rearrangement options also there, rearrangement. So the citrate converted to cis-aconitate with help of aconitase enzyme after removing of the water molecule. So here water molecule, you just compare these two, and COO, CH2, and here the HO is there, so HO plus H1 hydrogen. So finally we got H2O, H plus OH, and eliminating the water molecule, and finally we got the COO, CH2C, and COO, and COOH, and CH, and CH, and COOH, and finally the cis-aconitate, and this will be the reversible based upon the rearrangement. If you are considering like that, the water molecule we should be add, and finally we got the citrate like that. That is the reversible reaction. So now cis-aconitate, it is an intermediate compound where very quickly it will be converting here in this. Some of the cycles it may not be showing it, and sometimes directly the citrate convert into whatever the alpha get isocitrate, then alpha get like that. Some of the cycles there is no problem here. And here the cis-aconitate will be quickly reconverting. It is also reversible, and here that if you are considering from here to here or here to here, that water molecule involvement also there by the help of aconitase enzyme, and here also Fe2 plus involvement also there. Here also Fe2 plus involvement also there. This is also 6 carbons containing cis-aconitate, and that will be rearranging, converting to isocitrate. So here is the COO, CH2, CH2, C, and CH, and COO. So here you see that if you are adding water molecule, so if you are considering from the cis-aconitate to isocitrate, we should be add the water molecule. If you are considering isocitrate to cis-aconitate, because of the reversible reaction, we should be showing like this. Otherwise, if you are coming cis-aconitate to here, we should be add the water molecule. So that's why you see that here that water molecule extra here, that here COO, COO, CH, CH, HO extra adding here because of the water adding, and one more here the CH2, CH2, COO. So rearrangement. So after adding the water, finally we got isocitrate, and here also aconitase enzyme and the Fe2 plus involvement also there. So then after isocitrate converted into an oxalosuccinate, it is also irreversible reaction with involvement of NAD plus reducing equivalents. So that will be entered into electron transport chain, and after oxidation, and finally we got the ATP molecules. In each NAD we are getting the 3 ATPs. So here 2 involvements of the NADs if we are considering, and finally 6 ATPs we should be calculate. And generally in each Krebs cycle, whatever the one term, 12 ATPs we are getting, and if you multiply with 2, because of 2 molecules, if we are considering because of the 2 acetyl molecules we are getting from the pyruvate, so that's why 12 into 2, and finally we got 30 molecules in the citric acid cycle, and the glycols is 6 or 8 based upon the citric pathways. In the previous video I discussed it clearly, and so that's why 38 or 36. Finally we got this glycols as well as the citric acid cycle after oxidation of completely FADs, NADs, and direct production of the ATPs if you are calculating. And here that isocitrate converted into a reversible rate oxalosuccinate. Here the NAD converted into H plus isocitrate dehydrogenase. The involvement is there. You just compare the structure COO, CH2, CH2, and CH, COOH. So here extra ketone group here we are showing. So here hydrogens extra is this will be NAD accepting the hydrogen and converting the NADH plus H2 plus. So finally CO is there, that will be ketone form, C double O, and COO. Very clearly, crystal clear we are getting, and so finally we are getting oxalosuccinate. So that is also irreversible reaction. Isosaccharic dehydrogenase will be the enzyme. So now oxalosuccinate converted into alpha-ketoglutarate with the help of carboxylase enzyme by the removing of CO2 and Mn2 plus also involvement is there here. The CO3 here is the COO8 form. So here compare the COO, CH2, CH. So here there is no any CO group here, COH group. So that will be eliminating the form of CO2 by the enzyme, carboxylase enzyme. Along with Mn2 plus also required acting as coenzymes, and C11O, COO, and finally alpha beta glutarate. So this will be the COH group, functional group here. So alpha beta gamma delta lyset. Alpha keto group is the alpha carbon atom. So alpha ketoglutarate 8 form we are showing. That's why alpha ketoglutarate. Now comes to the alpha ketoglutarate converted into and succinyl CoA. So here alpha ketoglutarate dehydrogenase enzyme involvement is there. And NAD plus reducing equivalent also involvement is there. NAD plus and converted to NADH plus H plus, Ng2 plus also required. CO2 will be eliminating, and that is the oxidation, and the final water molecule also liberating. And so here that this alpha ketoglutarate acting as intermediate compound for the production of the glutamic acids also. Whatever that amino acids, glycine, alanine, valine, leucine, isoleucine, tryptophan, tyrosine, aromatic tyrosine, tryptophan, proline, aspartic acid, glutamic acid, glycine, all these the different kinds of levothyroxine acids are. So among those, so here the alpha ketoglutarate will be helpful to produce that glutamic acid. It is a reversible reaction. So here if you compare with the alpha ketoglutarate to succinyl CoA, so here you see that here COO and COO, CH2, CH2, so C11O, and S CoA will be adding here. S CoA will be adding coenzyme SH. So here and so here the finally NAD requirement also you see that coenzyme SH will be adding here. So that will be attaching to S CoA. So here the hydrogen bond, you should be remember that sulfur related. So now by the enzyme alpha ketoglutarate dehydrogenase. So now succinyl CoA we got succinyl CoA. So succinyl CoA, why we are calling this is succinic related structure CoA, succinyl CoA. The succinyl CoA, this is 4 carbons, and 1, 2, 3, 4 carbons along with S CoA, coenzyme is there. So the succinyl CoA converted into succinate. So what are the involvement is there? Here GDP involvement is there. Guanine diphosphate converted to GTP. So here GDP combining inorganic phosphate, we got the GTP. The GTP also here one more ADP accepting one phosphate and converting to GDP, and finally ADP converted to ATP. So that's why here either GTP or ATP we are showing, and here 2 cycles we are considering, and 2 ATPs should be 2 acetyl CoA involvement. So that's why we are getting here 2 GTP or slash 2 ATPs. So, whatever these are all whatever that triphosphates. So energy getting. So here now substrate level phosphorylation. Here this one, and this will be the succinic thiokinase enzyme. So because of that sulfur related involvement is there, sulfur related. So that's why the succinic thiokinase enzyme involvement is there. Here the substrate level phosphorylation. So we are getting the GTP or slash ATP or the triphosphate, and so that will be converted to succinate. So you see that here the COO, CH2, and here the involvement of this C11O rearrangement is there here, rearrangement, and see the CO and coenzyme SH will be eliminated. And here that after formation of this succinate and after the formation of coenzyme, just I am showing like this, you see that the coenzyme SH will be eliminating. So whatever present here S CoA with the hydrogen bond, so CoA SH breaking, so CoA SH will be eliminating. So finally we got the succinyl CoA to succinate with the help of succinic thiokinase because involvement of the S CoA enzyme is there. Sulfur related. So we are getting the GTP or ATP, 2 molecules. Now succinate converted into fumarate. Succinate converted into fumarate, and FAD involvement is there. Flavinadenine dinucleotide, FAD converted to FADH2, and 4 ATPs we are getting. Here the FAD, each FAD we are getting 2 ATPs if we are multiplying with the 2. Because of the 2 acetyl CoAs we are getting in the citric acid cycle. That's why the 4 ATP. and we should be consider the succinate converted to fumarate here. FAD involvement is there. You see that here carefully here, FAD accepting the hydrogens here, oxidation process purpose, the COO, and CH2 is there. Here one hydrogen accepting, here also one hydrogen accepting, FAD converted into FADH2, and finally we are getting that CH2, COOH, CH2, CH2OH. So finally we are getting the the fumarate. So then fumarate, it is a 4 carbons containing structure after adding the water molecule here with the help of fumarase enzyme. And so here the enzyme, you know that whatever the succinate converted into fumarate with the help of succinic dehydrogenase. It is there. And one more special here, whatever the enzymes are all are present in the matrix of the mitochondria except this succinic dehydrogenase, because this will be present in the inner membrane of the mitochondria. This is another important enzyme present only in the inner membrane. Rest of the whatever enzymes present in the matrix of the mitochondria, this is accepted. This succinic dehydrogenase, this will be present in the inner membrane of the whatever the mitochondria. So now let's come to the further structure. The succinate converted to fumarate. Now fumarate, this will be the structure COOH, CH, COOH after adding the water molecule with the help of fumarase enzyme. And finally we are getting the malate. You see that adding water, what are the rearrangement? COOH. Same, so here extra one hydrogen adding here CH2. So here there is no OH group here, but here the H2 was split up H plus plus OH minus. CH will be adding here second carbon, and OH will be adding here that third carbon. So OH we are getting here extra, and the COOH common. So finally converted that whatever the fumarate converted into malate with the help of fumarase enzyme. So here the fumarate also 4 carbons. And finally here the COOH, CHOOH, CH2COH. This is the structure of the malate. Involvement also there, NAD plus nicotinamide adenine dinucleotide involvement is there. 2 molecules. If we are adding the final, we are getting the 6 ATPs, because each and every 3 ATPs we are getting, and with the help of malate dehydrogenase enzyme. So here that malate converted into oxaloacetate. You just compare that together. COOH, CH2. So here NAD accepting that hydrogens, NAD accepting hydrogen and converting NADH plus H plus, and NADH plus H plus, and whatever that we are getting like this. So you see that here that NADH plus NAD, and like that NADH plus H plus, and finally this will be accepting by the NAD and converting NADH plus H plus, and you see that structural CO is the remaining C double ketone group, and COOH, COOH. So oxaloacetate we are getting. This is also 4 carbons. So 1, 2, 3, 4 oxaloacetate. Once again, this will be also helpful. It is a reversible reaction, useful for the one of the important amino acid production, aspartic acid. So here you see that oxaloacetate will be for aspartic acid reversible reaction. We are getting the alpha ketoglutarate. From that we are getting glutamic acid. This is also acidic containing amino acid. This is also aspartic acid, acidic nature containing amino acid. And here also the fat, from that also we are getting that after beta oxidation, degradation of the length of the fatty acid, converted to simple to acetyl-CoA, 2 molecules, and converted to acetyl-CoA, and finally we are getting the acetyl-CoA. So this is the central part of the acetyl-CoA we are getting from the fat metabolism as well as that protein metabolism. Also even also protein is nothing but a combination of the amino acids with the peptide bond by the elimination of that water molecules, and the COO-NH we are getting one amino acid of the carboxyl combined with another amino acid of the amino group after eliminating of the water molecule, and finally COO-NH we are getting. That is nothing but peptide bond. All the different kinds of amino acids linked with the peptide bond, whatever the 20 liver amino acids we are observing generally in the protein, and we are getting the protein. So here the degradation time, the protein oxidative catabolics and process, the degradation of the length of the protein molecule into simple amino acids, the oxidative catabolism converted to amino acid, and finally we are getting alanine. Alanine also will be helpful to produce the pyruvate. So here it is an interlinking process, total the citric acid cycle, and here the total by here acetyl-CoA combination, 3 NADs molecule involvement is there, 1 FAD molecule involvement is there, 1 GDP involvement is there, 2 molecules of water molecules. Also overall here reactions input, what we are getting the output. So here the 2 CO2 molecules, 3 NADH plus H plus and H plus, and 3 whatever that here already there, 3 NAD plus H plus 3 and FADH2 and GTP and coenzyme A. So here this is the Amphipathic pathway, and provide an intermediate for synthesis of amino acids and aromatic porphyrins, and that will be useful for the synthesis of chlorophyll also. And here one turn of each one we are getting 12 ATPs and 12 ATPs here only, 12 ATPs only here. And here also 6 we have to add. So to 12 into 2, 24 plus 6 we have to add. Finally and 30. So 30 from that citric acid cycle, including of the pyruvate acetyl-CoA 6. Here also 12 into 2 to 24. So here also 2 molecules with 6. So 24 plus 6 is equal to 30. In the glycolysis based upon the citric pathway, we are getting 8 or 6. The total we are getting 38 or 36, including of the glycolysis as well as the citric acid cycle. So here that maximum here the irreversible reactions are there, and here the simple rearrangement here intermediate compound only that will be interchanging, and there only that reversible we are showing. Sometimes directly also they will be showing there is no problem at all, no issues. So this will be the very important to production of that ATPs, and here also substrate level phosphorylation we are observing here also because of the production of the GDP into GTP, or we may showing ATP also. And here rest of the FAD is flavinoidine dinucleotide, nicotinamide dinucleotide. They're all oxidation process after entering to electron transport chain that we are producing with the help of oxygen, producing water molecules as well as that ATPs also we are generating. So this will be the actually taking place in the matrix of the mitochondria in the eukaryotic cases, whereas in the prokaryotic cytoplasm only cases because of the lacking of the mitochondria. So all these information very important. All competitive exams will be very helpful. Thank you very much.